Hello everyone, and thanks for tuning in to this webinar. It's an introduction to behavioral research methods. My name is Brian Sullivan. I am a Marie Curie experienced researcher here at Tobii, and I'm going to be talking about quite a few different topics within behavioral research methods, and it's intended to be focused on people with rather sort of introductory experience with experimental methodology. So we're not going to be talking anything in particular that would only be particular to eye tracking. This is sort of just for behavioral research methods in general, and these are kind of different tips and standard practices that can be used in a wide variety of experiments and not only eye tracking. So before we get underway, there's some webinar instructions. So please use the chat function in GoToWebinar if you have any questions that you'd like to ask. You can also use, there's the questions function as well. And additionally, you can listen to the presentation either by using your computer speakers or by calling into the conference number in your country. And please see your registration email for any more details that you might need. Now, the way things are constructed here, because I'm giving the talk, I'll probably just leave all questions for later at the end. The way things are set up here, I'll talk for about 45 minutes to about 60 minutes and then have time for questions, and I'm happy to answer anything that might come up as we're going through this presentation. So in research, you can't just wing it. There's a lot of different issues that might come up when you're doing experiments, and if you don't make enough plans in advance, you might get some unexpected results in the end. And so within behavioral research, there's some general steps that are sort of the norm that everyone practices. So the scientific process is a systematic inquiry to gain knowledge about the world. And so this is something that people have been practicing for quite some time now. And so we have general steps about how do you go about practicing the scientific method. Unfortunately, the real world is a bit messy, so you have these well-defined steps that work in general, but sometimes you have to use your judgment and opinions to kind of figure out the best way to address complex situations. And but I'll keep this talk mainly sort of at an abstract level to kind of keep it not to any particular experiment, although I'll bring up some examples every so often to try and ground things for you. So when you initially start out with before you might even have an experiment in mind, you might just have some sort of question and maybe a hypothesis about something in the world that you're interested in and how that relates to maybe some sort of drug treatment that you might give someone or some sort of stimuli or teaching process or something like that. And once you've come up with this question and hypothesis, there is some degree of research and review of prior literature that you should do beforehand before you even start doing any sort of inquiry yourself where you're actually doing an experiment. You can look and see what others have done and try and learn from them so you can know what to do and find out if people have already done what you may be trying to do. Additionally, once you've found out the lay of the land, you'll have an experimental method that you'll need to decide upon, and there's a bunch of different elements to this that I will go in depth about as we proceed in the lecture. Finally, after you've determined this particular experimental method and how you are going to run subjects and collect your data, you'll have a whole bunch of data at the end. So you'll have to analyze it somehow and then try and make sense of the results. And once you've done that, ideally you have some sort of report or write-up, some way to transmit that information to other individuals. Now, when you are designing a study, there are many general issues that come up. So there's basic things of what do you want to know about the world, and without initial questions you're probably not going to really have an experiment in the first place. So you have to find, given that you'd like to know something, what is it that you want to achieve? And In particular, if you're saying that you'd like to do an experiment, if there's something, say, about how do people look at pictures in the world or something like that, how will you answer your questions? So you can start with something that's kind of general, but then you have to progressively get a bit more specific. So in particular, you might have something of, well, I'm interested in if people look at certain things in images more. Maybe do they look at faces more than other things? So you have to start thinking about, if you have a general question, exactly how will you evaluate whether it's true or not. And then given that you have some way to measure it, you'll have to take whatever findings that you might have to do statistics or some other data visualization or something like that, so you can actually say something about it and relate it back to the initial question that you had. And then finally, given whatever you've learned, you need to be able to have some sort of recommendation. So did the experiment actually work for you? Maybe you need to run a second version of the experiment to improve on some things. And then, of course, too, you want to be able to act on the information. So did you find out something that's maybe medically relevant that should be transmitted to patients, or maybe you know something about consumer psychology that maybe would help developing products or something like that. Now, the experimental process has a lot of different steps, and so here I've just kind of outlined on a high level all the different things that you might be facing when you're thinking about starting an experiment. Now, this is a long list, and so I'm not actually going to be covering all of the elements here. So what I'm going to mainly just cover are sort of the initial process of deciding on a research question, thinking about the study design, and how you might prepare different elements with your subjects, and how you design different elements of stimulus presentation, as well as ordering of stimuli and things like that. With a little bit, depending on the timing here, I'll talk a little bit too about some of the actual experimental protocol when you're actually doing data collection, and some little bits on reporting eye tracking data. Now, when you start out, you need to define your research question. So everybody has little theories about the world and different ideas about things that are interesting to them, and so you have to start, potentially you just start with a sort of like a loosely formulated question about something in the world that interests you. Other times it might be motivated by prior research, or some other thing that you'd like to know about, maybe a disease or something like that. But for instance, we might take the example of maybe we're just curious, what do tennis players do with their eyes during a game? Well, that's a pretty general level question, and so if we want to make that into an experiment, we need to zone in a little bit more on what's important. So the general process here, so you start out with a sort of a vague question that is pointing you in the right direction but doesn't say exactly what you'd like to know. And so this process is called I always screw up this word, operationalize the question that you have. And so this is the process of going from vague to precise. So you have some sort of empirical measure that you'd be interested in that will relate back to whatever your initial question was. And so, for instance, asking what do tennis players do with their eyes during a game, there's a lot of different things that might mean. So maybe we want to zone in a little bit more, and we say, do tennis players keep their eye on the ball only on the ball during a serve when they're receiving? So now suddenly this becomes a bit more concrete, because then we can say that we want to gather empirical data, and we're going to say that visual attention as measured by the location of the fovea over time, so what an eye tracker measures, that is what we're interested in terms of how well visual attention is directed towards the ball. And so what you typically end up doing with whatever question you might have is to go from, again, something that's a bit fuzzy to something that's a bit more refined. And oftentimes along the way you might realize that certain ideas are too complex or expensive or just unfeasible if you haven't done prior research on that particular topic before. So for instance, with this setup, maybe it's not actually realistic for you to bring in tennis players or to go track tennis players and see what they do. Maybe you just really want to know just how do people track moving objects with their eyes, and you could do that in a simple lab setup that doesn't actually require getting tennis players and all these other things that might be required. Now, when you are going through this process of refining what your question is, a good thing to keep in mind is your experimental aims. And so this is something, if people talk to you, if you're writing a grant or doing some sort of project proposal, people will often ask you, what are your experimental aims? What are your specific aims? And so the general idea is that they are, on a general level, what you'd like to achieve and what you'd like to find. And what you can do is that, given that you have these aims, you translate them into specific testable hypotheses that you'd like to have something to say about the world. And so in general, it's good to have a sort of a high-level understanding of what is it that you're actually trying to measure, what does it say potentially with respect to background literature or theory. And so once you have this sort of high-level specific aim, you can zoom in on the more specific methodological issues of actually how are you going to carry it out, what are you going to measure. Now, when we're thinking about these sort of things, and along the way I'm just going to give a bunch of different nomenclature that people use within psychological testing, just to make sure that you have the background to know what people talk about when they're talking about their experiments. And so typically people will say that before you start an experiment, you should develop a hypothesis. It doesn't always happen that way, because sometimes things come up that you learn as you're doing one experiment and some new question arises. But the general idea is that if you're interested in a particular thing in the world, you want to have some idea about if there's a situation that you could alter, how would your alteration affect the outcome? So it's basically some sort of causal claim about how the world works. So a hypothesis will typically relate, say, two variables or measures, and it's a prediction on the relationship between them. So to people, what they'll typically call these is there's the independent variable or the predictor, and this is typically what the scientist is going to manipulate, and then there's the dependent variable. So this is the thing that depends on the independent variable, so it's the outcome of whatever you're going to measure. So for instance, we could be in a situation where I might make the claim that coffee increases saccadic accuracy. So this is something that could be empirically tested, and what that means here is that we could expose people in different situations to whether or not they had coffee or not, or maybe we gave them caffeinated versus decaffeinated, something like that, and then we could measure saccadic accuracy. And so we're saying that the outcome, the dependent variable saccadic accuracy, is related to how much coffee the person has consumed. And so these are the typical ways that people think about the things in the world that you might be testing. So you have something that you can manipulate, and then you have something that you can measure. And so all of this, the goal here that we want, what we want is to have scientific results. So we're interested in some sort of relatively deterministic system. Things in the world aren't always exactly deterministic, at least in the way that we measure them, but what we'd like to do is to have some sort of causal chain to say that when I give someone A in the world, B always happens. And so we're looking for these explanations about what goes on in the world, and this is why the scientific process exists the way it is. So we want to have objective evidence for saying that something does or does not happen. And so we want something that's replicable, precisely described, data-driven, systematic, and unbiased. These are all the goals that we have, and so we have to start thinking about, initially, before you even design the experiment, exactly how do you go about getting things correctly tuned so that you can actually do it in a scientific way and you're not biased. And so there's always a difference between, say, a person making a claim about a survey that's just on a website or something like that, how much do you like a particular thing, versus someone actually doing it in a lab. So there's lots of different variables that can affect outcomes, and so just because you can measure something doesn't always mean it's objective, and that's something we'll talk about a bit more. Now, when you're engaging these things, you have to think about the type of measures that you're going to collect. And so, of course, here, most likely in mind, you have something about eye movement measures, so maybe you're interested in fixation durations or latencies of saccades or something like that. But of course, there's lots of different things that you measure in the world, as well as the fact that you might have other things that you're measuring in addition to eye tracking. So maybe you're doing something with EEG or heart rate or something like that. And so when you're engaging in an experiment, you have to think about there are variables and measures, and then there are constructs. And so the general idea would be something like, take for instance the blood concentration of a protein. Now that is a variable. It could be high or low depending on the individual or something, but you could at any point in time actually measure it and be able to say something about the current level of it. Now the thing is though is that maybe you have some broader explanation that relates the protein to maybe something like mood or weight gain or something like that. So this would be where the idea of a construct comes in. So you measure something, but that measure is actually related to something that's a bit more complicated. And so sometimes what you're interested in may be quite simple, like maybe you only have a question about height, and it's relatively easy to measure that. But certain things, especially in the psychological realm, you might have something like someone's intelligence, their personality, which there's no easy way to directly assess that without using a bunch of different pieces of evidence. And so when you're designing an experiment, it's good to think about what measures you're going to use and why are they important. So you might have something like quantitative or qualitative measures. So maybe you're giving someone an application for a phone to test, and so you could ask qualitative questions of just how good did it seem to them, maybe were they fast or slow, did they seem to find things, and it's just kind of a very cursory level of analysis. Or you could do something where you actually measure quantitatively things like reaction time or fixation durations. And again, it really depends on what you'd like to know, and it has to do with how does this measure relate to whatever you want to find out about. So something like fixation duration, maybe you have a hypothesis that it's related to processing time or difficulty. So it's not necessarily that fixation duration in and of itself is what you're interested in, but it's because it relates to some broader context that gives you some idea about the human behavior. And so whichever measures you choose for whatever type of experiment that you're doing, you have to keep in mind that your measures need to be reliable and valid. So we'll talk a bit more about it, but the general idea is that you need something that other people can use and use in a way that isn't very idiosyncratic, so that whatever type of experiment that you do can be done elsewhere with very similar results. Now, before I get into some of the specifics about experimental design, just a quick little tour of the prep work that you should do before you actually engage in your experiment. So the literature review is something that's quite important, and it oftentimes can be a little bit daunting, especially if you're not familiar with a particular field. But the thing is that if you have a particular research problem, you need to know what you're studying and why. Why is it important? And in particular, what have other people done? Sometimes there are these unique cases where you're looking at something that's really never been done before, but most likely somebody's thought of something rather similar to what you're doing. And you can learn quite a bit because there's a massive amount of literature that's out there that can help you and importantly to avoid some of the pitfalls of doing bad experiments or having certain things that hadn't worked, or maybe learning something new that will inspire you to something else. So taking into account previous research and current research, as well as one of the most important things is that oftentimes in psychological research there can be a lot of findings and very particular experiments, but maybe not so much theory. So if you read up, you can find about what are the theoretical motivations and some sort of conceptual framework for thinking about why are people interested in this or that, because only looking at a couple experiments just to see the actual methods of how did they measure, say, fixation duration or fixation latency, something like that, that might not really tell you about what sort of cognitive mechanisms people are proposing. So you have to both look at kind of current research as well as maybe reviews or theoretical papers, and that will very much help you figure out how to design your own experiment. The other thing too is that especially now we're in an age where a lot of the search that used to be quite laborious is relatively easy, although online search covers a lot of things. There are still some things that you need to go to the library for, but at least in most cases you can find out whether it's in your library or not. And so if you're not aware, there's many dedicated sites that exist for getting research literature. So one of the favorites of mine is PubMed.com, and another one is Scholar.google.com. So PubMed is actually sponsored by the U.S. National Institute of Health and hosts a huge amount of research. It's not exclusively to the U.S. It covers topics around the world, although it's mainly in English. And you will find most of the abstracts for articles, plus sometimes the full articles, or at least links to the journals. Scholar Google is kind of similar, but now it's using the power of Google. So you have personal sites for researchers and can also sort of do a combination of searching personal sites for the research as well as sources for PDFs and things like that. There are other options here too, ResearchGate, Archive, Academia.edu. These are all good resources, so use them. You also, of course, have the journal websites themselves if you have a subscription or are interested in purchase. And additionally, too, there's something that people don't often do is to contact the authors. Now, right, so this roughly gets us back to where we were. And yeah, so, and again, sorry for not realizing that everything was muted. So now that we're back, so here there's multiple types of ways of thinking about experiments, and one way to think about it is in terms of basic research and applied research. So you have potentially research that's theoretically motivated or looking to improve things where there's a knowledge gap. You also have applied research where maybe there's a particular problem or an intervention that you'd like to try out on a particular population. Now, with other types of these research, typically it's going to take many subjects and trials for you to get all of the information that you need to say something about whatever it is that you'd like to study. However, it is good to keep in mind sometimes case study is the only thing that's possible. So maybe you might only be able to get access to one or just a few participants. So it might be something where a person has a very unique disorder or maybe a unique ability or something like that. Or maybe it's something where you'd like to study someone over several years or something like that. So it's rather difficult to keep lots and lots of people in those situations. Now, when you're thinking about experiments, and this is especially true with eye tracking studies because there's quite a bit of variety in the ways that people have approached looking at eye movements. And so the most common and well understood is in controlled experimental environments. So this is kind of like the typical lab situation. It doesn't always have to be a lab, but the general idea is that here you're in a situation where you're controlling almost all of the variables in the scenario. So you bring in everybody into the same room. They always look at the same screen. They're always presented with the same images or whatever you might be having people look at. And this is a way that you minimize a lot of the variables. And so generally speaking, the whole idea is that you have a variable that you manipulate as a researcher, and then if we can go with the assumption that everything else is remaining constant, then we can say if we have two different groups that were exposed to two different stimuli or something like that, we can make some comparisons, say the stimuli does something in one group but not in another. Now the thing is though is that in these controlled experiments they don't necessarily represent what's in real life. So in particular with having people view things on computer screens, well, a lot of human vision is actually, in the 3D world. So although people do look at screens and interact with things, maybe you're actually more interested in something a bit more natural that isn't just something that can be done in kind of like the typical lab setup. So that's where field experiments come into play. And so the general idea here would be that it's something that you're still controlling, but it's a little bit more real life. And then sort of the other extreme of that would be something more naturalistic and observational, where you have a study that maybe it's almost free form. You might have some independent variables that you try to control, but even then you're not going for total control. So maybe you just want to sort of understand how people really work in situations that might be difficult otherwise to access. So one of these things to go along with the tennis, the idea that I had before, is that you might be, so going along these lines, you might study smooth pursuit of objects on a screen, and that might teach you one thing. But then, well, what does it tell you about 3D objects? Well, let's look at smooth pursuit while receiving tennis volleys from a machine. Well, the machine keeps everything constant, and that's very nice. It's more controlled, and maybe now it's a bit more true to life, but that's still not what a real game is like. So then maybe you want to do mobile eye tracking with a person actually doing a full game. And there you won't even actually have very much control over any of the timing or anything like that. So that's one way to think about these different levels of experiments. Now, of course, too, in addition to different types of experiments, there are different types of measures. So here, and you can check this out later on if you're not familiar with this, but the general idea is that there are nominal, ordinal, interval, and ratio scales. They are basically just changing the level of granularity that we can make claims about things in terms of how do we categorize them. And so certain things, don't necessarily lend themselves to so you have something like measuring a height. That could be a ratio because it's either, you could be have no height at all and be 0, or you can be something that's quantifiable with a measuring tape or some other system. Whereas some things, say for instance, nominal scales with this situation here with AM, PM, you have this general idea. They're two categories, and without additional information, you don't really know one is bigger than the other, one has any ordering facts. So the general idea is that there's different types of measurement, and what is most important is that when you are measuring some things in the world, it's good to think about how you go about this, because this can change the type of statistical approach that you use or what methods you are using, because if you're looking at surveys, maybe you're doing something that's closer to nominal or ordinal scales, whereas something like fixation durations are going to be probably closer to ratio. Now, additionally, given that you have these types of measures that you want to have something that you affect on the subject that's in your lab. And so there's lots of different ways that you might alter variables. And so you could manipulate the context or the stimuli. Maybe you have different tests or instructions. There's all different ways to carry out experiments where you have a treatment group versus a no treatment group, stimulus 1 versus stimulus 2. And so this will always depend on the particular question that you have in mind. But one of the main things to think about, especially when you're starting out, is to think about what are the resources required, is it feasible, and think about the time, the complexity, and the budget that you might have, because certain questions, maybe there's a fast way to answer a question and not to do in an exhaustive manner that would be really expensive. Now, within psychology, there's design terminology that people use, and so you might hear people say something about one-way to n-way design. And so the general way of thinking about this is that when I'm saying 1 to n, that refers to the types of conditions that I might have, and additionally, how many variations within a particular condition I might have. So just to give an example, maybe we're interested in something like we want to relate reaction time to a visual stimulus, and in particular we're interested in age. So age would be, this is sort of like an independent variable that it's not necessarily we're not actually making people older or younger, literally, but we can separate groups out and then analyze our data after the fact. So we might have young or middle or older group. And then we could say something like, well, we measured the reaction times, and then we can do a bunch of different types of pairwise comparisons. So we might have something where if we have these 3 different groups, so this is a one way, so the one dimension that we're interested in is age, and then within age we have 3 different groups, and then within those groups, if we wanted to make all comparisons possible, that would be 3 comparisons. Now, just to illustrate how sometimes the complexity of experimental design can balloon, and this is why it's always good to think about these things beforehand in terms of how feasible is it, is this example here with a four-way design. So say we're still interested in reaction time, but now we're going to take into account athleticism, age, handedness, and gender. So now we would have something like and this in the nomenclature would be considered a 2 by 3 by 2 by 2 design. And so what that would mean is that we have people who are athletes and non-athletes, so that would be a 2. Then we have young, middle, and elder, that would be 3. And then we have people who are left-handed versus right-handed, which would be a 2. And then we'll have male or female, so then that's another 2. So with this four-way design, if you actually just multiply those all together, you realize that we have 24 different groups. So that's quite a few groups. So we went, if you consider the other situation where maybe we wanted to get 10 people in each condition of young, old, and middle, that might have been if we had 3 pairwise, if we have 3 groups, that would be maybe 30 people total if we wanted to run just 10 in each group. But now, if we wanted to have 10 people in each group here, where it's like we want an athlete who's young, who's left-handed, who's female, and we need 10 of them, then that's going to mean that we have 24 groups times 10, so quite a few more subjects. Additionally, and a very important one, especially when you come into statistics, is that also balloons the size of comparisons that you have. So it goes up from 3 comparisons to now 276. And so there's some major issues within statistics in terms of false positives that you can get. So you basically have to either plan your comparisons ahead of time or do what are called corrections for multiple comparisons. But these are things to think about when you're designing, and the general idea here is one that's pretty common. So be realistic and keep it simple. Lots of conditions are going to increase the complexity, and it's a lot easier in general to do a simple experiment that's easy to control than a very, very complicated one. Additionally, another element too, and this will always depend on the exact experiment that you're doing, but a control condition is usually crucial unless it's something that's, there's some situations where you can't always make a perfect control, but the general idea is that if you're giving, say, a person a drug and you want to see what happens with the disease, you typically want to keep in mind what people would say sort of like the placebo effect. So you give people a sham or a null condition where they think they're receiving the treatment, but they don't actually get it. And so sometimes, you know, in visual studies, that can be, it's quite variable in terms of what the control can be. So sometimes that might be, you know, if you're looking at eye movements, you know, for particular images, you might have sort of like control images where, you know, if you're interested in what people do when they look at faces, maybe you have images that just have no faces, or maybe scrambled versions of faces or something like that. So you have some sort of nice baseline to compare against. So you can actually try and control for any sort of compounding factors. Now, this brings up one of the key points here. So when you're doing experiments, this idea of experimental control, you have to keep in mind that there are the things that you directly manipulate explicitly. So whatever it is that you're actually interested in, they're actually actively presenting to the subject, or potentially recruited people for specific things, like maybe a particular age or something like that. But there's also the subject variables themselves. So in any study that you do, each person that's coming in is going to be to some degree a bit unique, and you never know if, for sure sometimes, whether did your experimental treatment actually work, or maybe it was just because of a particular thing about this participant, or maybe there was just some sort of random fluctuation where they had a good day or a bad day or who knows what. So there's a lot of things to keep in mind, and the general idea is that there's these compounding variables where bad protocol, potentially order effects where people are learning something about the stimuli that you're giving, or maybe you're making incorrect comparisons where you're comparing old people versus young people when you should be comparing old people versus old people and young versus young. Another common thing is that there's experimenter bias, where sometimes you can inadvertently, it isn't necessarily explicit bias, but can try, you can actually influence subject behavior And there can also be other things too where, depending on how you've designed your experiment, certain effects may arise not from your treatment but more due to the fact that you put people in a particular task or something like that. So all of these things now, these are very general level warnings because there's not, it always depends on the particular experiment that you have, but the general idea is think about potential confounding variables ahead of time, and there's a few different ways in terms of standardizing procedure and counterbalancing and trying to do subject matching and kind of stepping through the logic experiment that can minimize these problems. So when you are implementing your conditions, typically what people will talk about are two different ways to go about it. So there's independent measures, and so this would be between group. So for instance, you have say two different conditions, and everybody, you have a group of 20 people who are exposed to condition 1, and then you have another 20 people that are exposed to condition 2. So what this does is, in some cases, maybe there's something about one condition that gives you information about the second condition, so they have to be exclusive. And so there's some good reasons sometimes where you have to have independent groups. And often, too, potentially this can shorten the amount of time that the person is coming into your lab because they only need to do one condition and not both. However, this will also mean that you potentially would need more people. The other crucial thing that's the biggest worry is that participants might be variable between the groups. So how can you be confident that all the people in condition 1, there wasn't some systematic bias that put them in that, and then condition 2. And so it's not actually that the conditions are different because of what you did as an experimenter. It's due to poor planning in your subject selection, and you basically have a confounding variable. So one of the ways that people control for this is to do something that's called a within group measure or repeated measures. And so the general idea is that in this case, say we would have 20 people who come in who do both condition 1 and condition 2. And so this way, in particular, what you can do is control for the fact that, say if you're doing something where it's a test, and each individual, some individuals might be bad, some might be good, you can say that, so a particular person, they might go from bad to pretty good, whereas someone who is already pretty good might go to really great or something like that on the test. And so you can actually look at the pre and the post effects for each individual to say how much did they change as an individual, and not saying, oh, just kind of lump them all together, and because there's some issues with statistical analyses that basically you can end up washing out effects due to the fact that you're basically incorporating noise across subjects in the independent measures style. So repeated measures is usually more desirable but not always possible. With both of these situations, because depending on exactly how you're presenting your stimuli, there's oftentimes repetition or sequence or ordering effects where you may have to make sure that the stimuli that you present, they are usually want to try to randomize, order some way, or have some sort of pseudo-random thing so they're not guessing things ahead of time, or maybe learning something. There's also some other issues too with potentially if your experiment's too long, they might become fatigued. Additionally, this might lead to some attrition, so some subjects can't always continue if they might have, they're getting tired, or maybe there's medical issues or timing, so you always have to keep these things in mind. Now, when you are bringing people in, and this is something I'd measured before, you always have to think about your experiment and the measures that you're using experiment, the reliability of them, and the general idea is just, will we get the same result? If another experimenter comes along, will they be able to get the same results that you do? And typically the best way to kind of ensure this is to have a very good experimental protocol and to have trained researchers performing the experiments so that everything is done in a very well-defined way so you're not leaving things to luck. Another thing too to think about, and this is more in terms of the logic of your experiments and what type of conclusions that you might want to draw, especially if you have some sort of long logical chain where you're, say, trying to relate something like fixation durations to something complicated like someone's depression or who knows what. There could be any number of things. And if you have something where you're going from one very small measure to something that's actually rather complicated, where it's like some aspect of somebody's personality, you have to think about all the steps in the logical chain to say, how are these different pieces related? And so people think about this validity in terms of how your measures might relate to whatever it is that you truly want to understand. So there's a couple of different ways that people put it in terms of face validity, where something just doesn't make common sense. It's kind of like a first measure. It doesn't always hold because sometimes things that don't make common sense are true, but you can kind of, if you're an experienced researcher and someone tells you that fixation durations are a function of the phase of the moon or something like that, you can sort of say, that's probably unlikely, just based off of common sense. The other thing, though, too, is that to think about content validity. Just because you measure one thing doesn't mean necessarily covers everything. So maybe you need to think about reaction times from hand movements or something like that, and not only eye movements. The other thing, too, is to think about, so there's another element is criterion validity. So it's talking about how predictive of something something is. So oftentimes in statistical tests, people will talk about, say, R-squared, a value like that, which tells you the sort of the predictive value of something that's correlated with another thing. And so sometimes things may be very weakly predictive or sometimes very strongly predictive. So, and that can have a major influence, because if you're measuring something and it only really weakly predicts sort of the construct that you're interested in, might not actually be very important. So you can help you think about things theoretically. And of course, too, again, and this goes along the sort of same lines of, if you can't make predictions, it's going to be quite difficult. It seems like you've chosen the wrong measure. And then you also, too, have to think about the construct. So it's like whatever it is that you're actually interested in, if it is something rather complicated, how does that measure fit in with the causal model or whatever prior work people have done? Now, another level of validity to consider, too, is that there's internal validity. So again, this all has to do with the logical chain that you might have. So if you have some logical inconsistencies or poor assumptions, you need to make sure that you've thought about why you've chosen one thing or another. And if you have something like for instance, maybe you're doing something with pupil size and you're saying, oh, pupil size changes as the effect of task difficulty, but then you didn't take into account that your stimuli when the task was difficult were bright and when it was easy were dark or something like that. So you have a confounding element that you didn't even realize. So this actually makes it internally invalid that you're making this claim that it's due to one thing when it's actually due to a totally different reason. So it's always it's not always easy to find out what the compounds are, but there are some things that are more obvious than others. And again, the general element is with good research protocols, if you write out things ahead of time, make sure that everybody who's running your experiments is very clear on what they need to do and very organized. That can help minimize some of these issues. Another thing, too, is the external validity. So if you have particular findings, can they be generalized to the body of work at large? Does it relate to theory? What results do you have? Would they replicate across other situations or other populations? So this, too, because if you're finding something that totally flies in the face of everything that everybody else has done before, most likely you're wrong, but sometimes you're not, and you found something that's really interesting. So if you are doing something that seems invalid, you just have to think about what is the reason, what's the logic, why is my result different than everybody else's, and think about some causal steps that would explain why you're getting one result or another. So I think here I'm going to kind of try to wrap up, just because so the webinar was just supposed to last to 6, and I want to make sure that giving everyone has enough time to maybe ask any questions. I'm going to very quickly go through the last few slides in about 5 minutes. There is a bit more here than unfortunately, given the delay in getting the audio working, there's a bit more here. So I'm happy to answer questions also via email, as well as the slides what I'll do is I'll go through the slides and have them in the video so you can review later on, because what we typically do is to send a link out to the video afterward, so at least you can review them if you have to leave now that the hour is coming up. So just to summarize this, and this is really the most crucial part of the seminar, but we're going to talk a little bit later about a couple other things. But the general idea is to look before you leap. Make sure you know exactly what your hypothesis is and what the logic is for whatever methodology you choose. You know, if you, in particular with eye tracking, you really have to think about what is it that you will answer with eye tracking, and it's not just because, oh, it's a cool toy that we have in our lab. You got to think about, you know, what are you measuring? How does this relate to prior work, or you know, whatever it is that you might be interested in, and thoroughly consider your design. A lot of planning ahead of time will save you a lot of pain later on, because experimenting typically involves a lot of trial and error. Most people aren't going to be perfect the first time because you're trying something new. It's so there's not, and it can be a bit unforgiving if you decide to run an experiment and then realize that you did certain things wrong, and then you've realized that you wasted time and wasted having subjects come in. So thinking a lot before you actually go through everything can be quite worthwhile. And again, and going along with this, to make sure to always pilot ahead of time, because just because you have an experiment that seems to work and maybe works on you and one of the other researchers that's in your lab or something like that, doesn't mean that it really works with the rest of the population. So it's always good to try out as many people as possible, and to you know, before you bring it up to the next level where you're getting in you know, actually subjects who are getting paid, who have to go through you know, whatever other types of testing that you might have. So it's always good to pilot things, and then in particular to look at your data ahead of time, because this is something that's quite common, where people get lots of data and then don't look at it as they're getting it, only to realize much later there's a mistake and they might have to throw it away, or just accept that there was something crucial that they missed. So now I'll just go quickly through just the last few slides here. So one of the other important things to think about are your subjects. So in particular, I mean, is because they are representative of the sample size that you're going to have. So it's kind of like a treasure hunt, where you need to make sure you have enough people to find what you're looking for. And when you're recruiting subjects, there's many different things to incorporate. Excuse me. So you have to think about the resources you have in terms of money, or what facilities the people might require. Is there any sort of risk that you might be exposing them to? How will you actually go about recruiting them? What if it's a particular group of people that you're interested in? You might have some assumptions about certain statistical tests that require, you know, more or less people to come in. You might have some other stuff too, with like type of stimuli that only you know, you're only interested in certain types of people, say only people who are colorblind or people who aren't colorblind. There's all these different things that you have to kind of think about beforehand, and especially too because bringing in subjects, that's going to be often like one of the more time-intensive aspects. It's something that you want to think about ahead of time. The other thing too to consider with sample size is that if you have some effect that's very small, if you're looking at something where you want to detect something that's on the order of tens of milliseconds, difference between people, you're most likely either going to have the people repeat whatever it is that you're having them do lots and lots of times, or you need to get lots and lots of people in. So either way, it means that you're looking at an investment in time and most likely money. So you want to try to make sure that you know ahead of time exactly what you're getting into. Additionally, too, if you have a very complex task with lots of factors, like I was talking about before, where you have, you know, going from a one-way design to a four-way design, that can dramatically increase how many subjects you might need. Additionally, too, if you have something that you want to say that's representative about the sample of people, then you're going to need more individuals to better sample the population. Similarly, too, there's issues with generalizability, so the more people that come in, the more you might have to say about how it applies to everyone. Now, one of the things here, and this is a bit of a complicated one that I'm going to go through quickly later on, which is in particular about the conditions in presentation in terms of counterbalancing. But for now, we'll just focus on sort of the ideal situation is that you would like to get people in the right place when you're assigning conditions. So you have a particular population that you're interested in. If so, how do you recruit them, and how do you make sure that they're representative and they're selected in an unbiased manner? It's actually rather difficult to do this because there's selection bias where some people want to be in experiments, some people don't. You can't actually be truly random, but you should try and make it a roughly equal opportunity if possible, and try and get people coming in who aren't a very specialized population, who don't actually represent the world at large. The other thing too is that if you're putting people in conditions, you shouldn't be biased towards putting them into conditions. So typically you'll have some sort of randomization procedure where you effectively just assign people at random, or there might be pseudo-random if it's dependent on certain things where they have to have certain qualifications of being in a particular group to be assigned to a certain condition. But in as much as you can, you try to reduce bias, so effectively there's nothing going on that might affect your results later on. Another thing to think about too, and this is in particular often with more sort of clinical and medically based studies, but if you need matching subjects, this is something where, say for instance, if you're interested in individuals of a certain age group or maybe IQ or socioeconomic status, this is something that you're going to have to plan for in advance, where you might need to actively recruit subjects from a particular walk of life, and you probably will need to do some level of testing before you can actually say whether they can come into your experiment or not. And that's it's just extra work that you need to do to find out if you're matching. But this can sometimes be quite crucial. For instance, maybe you're looking at children with autism, and it turns out autistic children on average sometimes have very high IQs relative to typical children. So then you might need to factor that in whatever experiment you do, where you need to get age-matched controls that also have similar IQ, and you can't just select any children just because they're convenient to get. Another issue here, and this is, there's a lot of different flavors of this, but the general idea with counterbalancing is that you want to get rid of ordering effects and sequence effects, where, for instance, say if you had a series of tests that you administered and people, for whatever reason, get better, but maybe their reaction time increases or the percent correct goes up. If you always present everyone with the same exact order of stimuli or conditions, you potentially have a problem where you can't say very much about the stimulus that happened at the beginning versus the end, because there's this confound where everybody's actually improving as the test goes along. So there's a couple of ways you can address this. One way is to totally randomize your stimulus, and this sometimes is necessary if you have a very large amount of stimuli or a large amount of conditions and it's not possible to totally counterbalance. But in simple situations, what you do is you typically have subjects are exposed to, across subjects, you expose them to different condition orders. So what I'm showing here in the bottom right is this Latin square, which is a very common thing that people will use, that allows you to sort of make sure that you have a balance without necessarily having to run every single possible combination of conditions. And so the general idea would be that you have, say for instance, with this 3 by 3 square, you have 3 different conditions, A, B, and C, and then you have this different ordering where the Latin square is basically you just write this matrix where you keep on shifting over one as you go down in the rows. And so then what you've done now is that you have different versions of the sequence of ordering. It's been balanced in a way that you can try and get rid of some of these issues with the possible ordering effects. Now, so the 3 by 3 doesn't actually incorporate every possible variation, because you could have other variants, but the general idea is that it's sort of representative of things balanced on average. So this can be quite useful because oftentimes you're not going to have enough subjects to run them in every possible permutation of things, so you have to do some subset. So counterbalancing in general is there's actually quite a few different ways to do it, as you can see here, but it's something worth looking into when you're thinking about stimulus presentation. And so one last thing too, but it's quite important, and hopefully everyone's aware within all types of science that involve people, good ethical practice is a must. And so there's quite a few kind of standard things that have been proposed throughout the years. So if you're really interested, if you haven't done this sort of thing before, there's this Declaration of Helsinki that I include this link to Wikipedia. And so the general idea is just to perform science in good faith without deliberately trying to deceive people unless absolutely necessary, and not putting people into more risk than necessary unless you absolutely need to, and with full consent from the individual. So generally speaking, you want people to know exactly what they're getting into, and not get into, well, situations that would be morally reprehensible or legally problematic. So what I'm going to do here now is just wrap up, and I'm just going to step through the slides, but I'm not going to introduce them because these are more about some aspects with the protocol and just some recommendations for basically just keeping a good notebook and being organized. So if there's any questions now, because we're a bit over time, where it's let's see, almost quarter after. So if anybody has questions or wants clarification, please go ahead and you can type that in to either the question form or the chat area, and I'll be happy to answer. Okay, so it looks like there aren't any questions here, so I'll just step through the last of the slides, and I'll put up some of my contact information that you can use to if you have any further questions. And right, this is another additional thing too. With the eye tracking data reporting, it's quite important to make sure that others can replicate your report. So here I'll just, so I see a question here, so I'll go ahead and answer that, but I'll just leave this up. So this is the final slide, so thank you for tuning in. You can contact me at brian.sullivan@tobii.com if you have any questions. I have a blog, Vision Research blog, that talks about some different issues with eye tracking and experimental methodology if you're curious. You can also contact training.analysis@Tobii for other inquiries. And of course too, so this is a lot of information, and typically if you're, say, in a psychology department, this would be a full semester course where we would get trained in experimental methodology and statistics and things like that. So for about an hour long, this is quite a bit to try and put into your into sort of like a very concise way. So by all means, look up for some follow-up information. There's Coursera, open course, other online courses. There's lots of different books for research methods in behavioral science. You can find them on online bookstores. And thank you again for your participation.